Welcome students. This is for fourth semester students of the unit 5, lecture number 3. We have already completed 2 lectures and this is lecture number 3 for an interesting topic: natural allergens, classification, preparation and standardization of allergenic extracts. So it's an interesting to know about the natural allergens and some of the example natural allergens which we are coming across in our garden and in our home. This we have taken some examples of plants. Apart from that, you should know that there are questions which they are asking: how you are standardizing an allergens, and characterization of allergens is needed for the student should know. And you as a pharmacy student, you know some details about the histamines and antihistamines and its therapeutic actions and all these things you might have come across. So now here in this, according to the university syllabus, we are having this syllabus, that is study of biological source, chemical constituents, uses of drugs of natural origin containing following plants and drugs products. They did not give the list, but we have taken some few which we are coming across in our homely uses. And natural allergens, we should know what all natural allergens: classification, preparation and standardization of allergenic extract. So here the immunological reaction to an allergen, antigen, allergen, seen in a patient with defective immune system is called as allergy. The group of substances that causes allergic manifestation are called as allergens. Allergies are abnormal immune system reactions to things that are typically harmless to most of the people, but it is dangerous also. When you are allergic to something, your immune system mistakenly believes that this substance is harmful to your body. Substance that causes allergic reactions such as even allergens, certain foods, dust, plant pollens, or even medicines like antibiotics are known to as an allergens. In an attempt to protect the body, the immune system produces immunoglobulin E antibodies to that allergen. Those antibodies then causes certain cells in the body to release chemicals into the bloodstream such as histamine. Allergic reaction can be mild, like a running nose, or they can serve like difficulty in breathing or an asthmatic attack, bronchitis. Some types of allergies produce multiple symptoms, and in rare cases an allergic reaction can become very severe. This severe reaction is called anaphylaxis. Usually anaphylactic shocks can be happen in penicillin injection. Even we have seen many vaccinations, sometimes some people will get anaphylactic shocks. And even streptomycin previously, they also used to give sometimes anaphylaxis type of shock, which gives strong choking, breathing difficulty and swelling of lips and various rhinitis. All these things will happen, redness of the skin, rashes. So some of the signs of anaphylaxis are difficulty in breathing, difficulty in swallowing, swelling in the lips, tongues and throat and other parts of the body, and dizziness and loss of consciousness. Anaphylaxis usually occurs minutes after exposure to a triggering substance such as even a peanut. The tendency to develop allergies is often hereditary, which means it can be even passed on through your gene. Most common allergens. Number one, foods. We are taking many seafoods, tomatoes, many foods, they may have common allergies. Number two, insect bites and stings. Even snake bite, they will cause even stings from various insects. Airborne particles, environmental allergens. Like sometimes even from the mosquito, even from the bugs and small insects, sometimes from the flowers, from the garden, sometimes from various pollens, the dust which comes from the soil, medicines. Also they will cause common allergen. They are also called as sometimes allergens to cause. Number five, chemicals which we are using in our home, including detergents, washing materials, toilet cleaners, air fresheners, many chemicals which we are using. When we come to food, milk and other dairy products, eggs, wheat, soya, peanuts and seafoods. They symptoms like they will cause an itching, rashes, a stuffy nose and diarrhea. And an insect bite and stings, they may cause the venom poison, and the insect bites and stings will cause. Airborne particles, which we say that environmental allergens, dust mites, tiny bugs live in our house as a dust, mold spores, animals, dander, flakes of scaly, dried skin from animals and even by human, and dried saliva from your pets, and pollens from grasses and ragweeds and trees. When you come to the medicines as an allergen, antibiotics and many other medicines including over-the-counter OTC drugs, medicines, painkillers, and when we use chemicals, household chemicals, some chemicals of laundry detergents, dyes, household cleaners and pesticides which we are using, these all comes as an allergen to give the allergy for us. So the principal types of reaction observed in allergy. You can see many allergy means rashes, itching, redness of the skin, triple response, urticaria, lips swollen, eyes, base swollen, tongue swollen, throat choking, bronchus, breathing difficulty, ear scratching, gonadal scratching. So these all type of diarrhea, palpitation. So there are many urticaria, angioedema, anaphylaxis. These all are the main allergies in which type 1 reaction, immediate type, anaphylactics, the allergen react within these antibodies acting the cell like penicillin. When you inject somebody's allergy, they will get anaphylactic shock, acting the cell and causing release of pharmacological active substance like histamine, leukotrienes, etc. immediately and causing effects such as urticaria, anaphylaxis or anaphylactic shock and asthma. Type 2 reaction, autoallergy, where the allergen combines with the protein in the body so that the body treats it as a foreign protein and forms antibodies. Type 3 reaction, when antigen and antibody from complexes and activate the complement, starting an inflammation response. That is the third one. It will start an inflammation response. Type 4 reaction, delayed type allergy, in which antigen specific receptors produces the T lymphocytes, allergy like contact dermatitis. So, 4 types of reaction, principal allergy reaction the student should know. Treatment of allergy, number 1, first environmental control from where the allergy came, stop to eliminate or minimize the exposure to the allergen for the particular person. Second one, symptomatic drug therapy by the use of antihistamines, corticosteroids and sympathomimetics like adrenaline or epinephrine can be given. Specific immunotherapy is the third one. So, first one is eliminating the environment with minimizing the exposure to the particular allergen which caused the particular patient with an allergy. And second one, symptomatic drug treatment with antihistamine, corticosteroids or sympathomimetic likes adrenaline or epinephrine or any other sympathomimetic drugs. Specific immunotherapies can be given for specific poisoning, toxicity, allergy, antidotes. Specific antidotes can also be used. Allergenic extracts. When you talk about allergenic extracts, there will be allergenic extracts for various diagnostic purpose and to find out what type of allergy is present in the particular patient. So we should know that what do you mean by allergenic extract. Allergenic extracts are concentrated solution or suspensions of allergens used for diagnosis and treatment of allergic diseases. Even for treatment of allergic diseases it can be used. Usually they are designated or designated as being aqueous, or that means they are added with or they are made with or they are supported with aqueous or glycerinated products. Normal saline or a similar isotonic electrolytes solution usually used for their dilution. When you want to dilute an allergenic extract for various purposes like diagnosis or maybe for treatment, it has to be diluted with an electrolytic solution which is isotonic, or a normal saline can be used. When we come to an allergenic extracts available commercially in the market or in the field of our pharmacy, we should know what all the allergenic extracts available. Number 1, diagnostic mixture. Number 2, customer treatment mixture. Number 3, scratch testing mixture. Number 4, intradermal test extract. Number 5, therapeutic extract. Number 6, autogenous extract. Number 7, adjuvant extract. Number 8, standardized extract. So you should know that list out, classification, how many allergen extracts available commercially in pharmacy. Number 1, diagnostic mixture, customer treatment mixtures, scratch testing mixtures, intradermal test extracts, therapeutic extracts, autogenous extracts, adjuvant extracts, standardized extracts. These are the 8 samples, like types of allergen extracts available. When we come to stability and storage of this allergen or allergens extracts, which is used for diagnostic purpose in the treatment of allergy, usually they are glycerinated or aqueous, and lipophilized products are more stable than aqueous extract. Inclusive of tween 20 surface acting agents like tween 20, tween 80 or human serum albumin reduces surface absorption, thereby reduce loss of allergens. Adjuvants extracts should not be diluted with either phosphate buffered saline or coca solution, is a caution, since they release the allergen from the adjuvant. Instead, dilution with normal saline containing 0.4% phenol is suggested. All allergenic extracts should be refrigerated at 2 to 8 degrees centigrade, and freezing is to be avoided. The expiry date or expiration date of aqueous extract is usually 18 months, while the glycerinated, stretched test and the bulk extracts is 3 years, can be extended. Lipolyzed or lipophilic products, lipolyzed product of allergenic extract, the expiry date is 4 years or 18 months after reconstructions can be given. So some questions, if they ask about stability and storage of allergenic extracts, this is the thing which you should write. What all the advantage of lipophilization, advantage of glycerin, addition of glycerinized or glycerinated, and even what aqueous can also be used. What's the difference in their expiry dates, that means the stability, and their storage conditions cannot be kept in freezer. These all things should be mentioned by you. Characterization and standardization of allergenic extract. They are asking this question for 5-mark question. So we should know in vitro methods and in vivo methods. You know that in vitro, externally, in vivo is internal living organism. So in vitro methods, number 1, polyacrylamide gel electrophoresis, thin layer isoelectric focusing. Third method, immunoblot assay, competitive ELISA, that means enzyme linked immunosorbent assay. Number 4, crossed immunoelectrophoresis CIE. And number 5, crossed radioimmunoelectrophoresis CRIE. And number 6, high performance liquid chromatography HPLC. And number 7 it is. Number 7, radio allergens test RAST. So these all are the 7 in vitro methods available for standardization or characterization of our allergenic extracts. In vivo studies, histamine equivalent prick method HEP, and number 2, intradermal erythema method. So very interesting to know about in vitro method and in vivo method. We should classify very clearly how they are standardized and characterized session is done for an allergenic extract. Number 1, polyacrylamide gel electrophoresis, PAGE, polyacrylamide gel electrophoresis page. In the phase, in the page or the passage, not page, passage of sodium dodecyl sulfate, SDS, sodium dodecyl sulfate, polyacrylamide gel electrophoresis. You can see this picture, how the gel is detecting the migration of the antigen compound or allergen which you kept there. The various component of allergenic extract migrate on the gel in relation to their apparent molecular weight, and the gel is subsequently stained, colored to give an band pattern to identify as shown in the picture. The maker proteins of known molecular weight are run on the same gel to serve as a standard. You can say that is even a marker protein or a same protein or a standard protein which is responsible for that allergenic reaction can be taken as a standard for this polyacrylamide gel electrophoresis. Number 2, thin layer isoelectric focusing, IEF. Here you can see the anode and the cathode and how the isoelectric point, the movement of the component which you have kept, that is amphoteric sample component, that means deuterions. The various component of allergenic extract are separated to their isoelectric point. The sodium dodecyl sulfate polyacrylamide gel electrophoresis, SDSPAGE, and IEF, isoelectric focusing techniques may be combined to construct two-dimensional gel that provide detailed fingerprints, comparing with a marker, comparing with a standard in an area of fingerprints of the composition of allergenic extracts, including allergenic and non-allergenic proteins. And number 3, immunoblot assay. Here you can see that immunoblot or immunostaining of blood, how you are obtaining an autoradiography of antigen band of visualization. In this technique, the separated components of allergenic extract by phase, that means polyacrylamide gel electrophoresis, are transferred passively or electrophorically to nitrocellulose papers, which are segmentally reacted with immunoglobulin E containing sera and radio labeled or enzyme labeled, radioactive labeled or enzyme labeled anti-human immunoglobulin E to produce autoradiograph as shown in the picture, or colored or color banding patterns. Immunostaining of blood can be identified. Here only the allergenic components of the extract produce visible bands. The competitive enzyme linked immunosorbent assay, that means ELISA assay, also can be performed in immunoblot assay. Number 4, cross immunoelectrophoresis, CIE, the antigenic, heterogenic or allergenic extract is studied by conducting crossed electrophoresis on a gel containing rabbit antiserum to allergen in question, or in finding out or to identify. So you can do a first electrophoresis and then a second electrophoresis. After the electrophoresis, cut the strip and the gel and transfer, like how you are doing the two-dimensional cross immunoelectrophoresis can be performed. And cross radio immunoelectrophoresis, also CRIE, the gel containing unstained precipitin arcs as shown in the picture, obtained by crossed radio immunoelectrophoresis, are reacted segmentally with immunoglobulin E antibodies and radioactive or radio labeled anti-human immunoglobulin E to produce cross radio immunoelectrophoresis or cross radio immunoelectrophoresis radio autoradiograph, or the band can be obtained as shown in the picture. Number 6, high performance liquid chromatography. We know that it is in chromatography is a technique to separation of mixture of compound. So a preparative, a large quantity in the small HPLC, high performance liquid chromatography column, can get separated and we can detect by means of the detector, UV detector. HPLC is used in analytical scale to provide a rapid method to fingerprint with comparing with a standard allergenic extract, or it is used as a preparative for separating the quantity and separating the amount. And for the further analysis, preparative scale to isolate the allergen rich fractions can be done. And number 7, it is not 6, it is 7. I made a mistake while typing. 7 in vitro studies are there. Radio allegro or allegrosorbent test. Radio allegrosorbent test. RAST. Radio allegrosorbent test, RAST. We can have this test, especially RAST, the radio allegrosorbent test, is used as a diagnostic test to detect the presence of allergenic specific immunoglobulin E antibodies, and also to quantitate amount of allergen or total allergenic activity in a crude extract can be identified by this RAST. So the student should know that the quantification can be done by this 7th in vitro study. Note this radio allergeno or allergosorbent test as described by White et al., a researcher in 1967, was historically the first immunoassay available for the measurement of immunoglobulin E antibodies to specific allergens in human serum. So it's an interesting information I have given for you. Any questions which they ask, you should have an idea to write about in vitro studies. 7 tests are there. In vivo methods of standardizing allergenic extract, skin testing is the most widely used in vivo, not in vitro, in vivo methods of standardizing allergenic extracts, wherein biological proteins are assigned. Sometimes we used to find out the area by pointing out by rounding or putting one square, and the area can be determined. Histamine equivalent prick method, HEP. The patient sensitivity by clinical history and skin testing to the allergen in question to find out, that means in question to find out which one is not known, are tested by prick technique to various dilutions of the test extract and to histamine dihydrochloride 1 to 10 milligram per ml. The activity of 1 HEP. What is this 1 HEP? When extract evoke will equal the size, the swelling, redness to that produced by 1 milligram per ml histamine, and the potency is labeled in biological units, BU. As 1 HEP is equal to 1000 biological units per ml. Second one, only 2 are there for in vivo study. Intradermal erythema method. Here also the same thing. You can see the dermal injection, how they are injecting the allergen and the test dose and finding out the patient sensitivity by the clinical history and the skin testing to the allergen in the question, that means in finding out, or the sample which you want to test are tested by the intradermal titration to various threefold dilution of both the test allergen and the reference extract. The testing flare diameter are compared by parallel line assay as shown in the picture. From the above effort of standardization, it can be expected that accuracy of diagnosis and effectiveness of allergen immunotherapy will markedly improve. Some examples from plant source, some common plants causing allergies, which we should know that. Arnica. This is blackwood, very big tree, like acacia. Another species, Peruvian lily, and this is asparagus, which is very nutritive. Bulb looks like onion. Variety also same thing, asparagus officinalis. And this is a garden plant in our home, chamomile. They are very much highly allergenic. Fig tree and fig fruit. Mint, peppermint mint which you are using. Lavender flavor which you are using. They are also allergenic. Garlic and Ginkgo biloba is having a highly effective for various purposes. Nutritive, nerve tonic, aphrodisiac, very highly useful for infertility. And you can see this beautiful flower of daffodil in our garden. They are also allergenic. Lemon, kiwi fruit. So there are still many, but we have taken a few examples. If they ask allergenic caused by plants, example and detail what all the allergens present in them. If they ask, the student should be able to write. Arnica, Arnica montana. Arnica common name, Arnica mountain tobacco. It can be called mountain tobacco or wolfbane. Botanical name, Arnica montana family, Arteraceae, or it can come to an Compositae family according to previous. They have two species. This is Arnica longifolia. You have yellowish flower. It is an yellow flowered aromatic herbivorous perennial which tends to grow in meadows and light woodlands. The stem is hard, hairy leaves in basal rosette. It flowers in early summer. Uses, it is major source of tincture of Arnica, which is still frequently applied to sprains, pain relief, brushes and wounds, although the evidence of it being helpful is limited. Other species used for the tincture of Arnica include Arnica longifolia, Arnica chamisensis, and Arnica chalenensis. What is the allergens which is present in it? Arnica montana, the allergen present is sesquiterpenes, lactones, helanin, carbarone, 10 acetoxy 8,9 epoxy, thiamaliso butyrate. So this is the compound which is present in it, causes allergy. Allergy of Arnica, that means Arnica, not Artica. Arnica has been recognized for several centuries and over 100 cases reported in the literature which we have researched, we have seen. Most cases appear to be from self-treatment with tincture of Arnica. They are using the tincture without proper guidance of the physician. They are getting allergy. Arnica containing a group of chemicals called sesquiterpene, lactones, and notably helinalin, which we have shown there, and its esters. They are causing severe allergenic reaction, urticaria, redness of the skin, even bronchitis. So they are allergenic, which is present in them. Blackwood is common name Australia blackwood or wattle of acacia. It's a big plant. The flower looks like same. They are allergenic. Botanical name Acacia melanoxylon, family Leguminosae. Description as evergreen vallet with dense foliage, balls of cream flowers as shown in the figure, and twisted pods. Height is 20 meter. Uses gum arabic derived from acacia. Usually acacia senegal is which we are using for acacia, gum acacia, is used for printing trade and binding agent in the various pharmaceutical preparation, making some medications like tablets, granules. Because of its excellent timber properties, also it has been used for timbers, and blackwood is used making furniture, boats, musical instruments, etc. But they have some allergen. You can see the blackwood. Commonly it's available in our place and the forest area. Allergen present in the blackwood or the allergens appears to be 2,6-dimethoxy-1,4-benzoquinone, acamylin and melacascidin in heartwood of this, which causes allergy, hand dermatitis, and has been reported after contact with both wood and the gum allergy or Arabic. And even sawdust, when they are using as in furniture workers, carpenters, having in the problem, particularly in the furniture makers, hay fever, rhinitis, conjunctivitis, and other respiratory problems are of increasing concern. Peruvian lily. See that beautiful flower, Peruvian lily. Common name, Peruvian lily, lily of Incas, San Martin flowers. Botanical name is Alstroemeria and family Amaryllidaceae. There are different varieties of color from the different places. Description: a herbaceous perennial with fleshy tuberous root. They have twisted lanceolate leaves, leaves especially, irregular trumpet-shaped flower, which are bone or slender, leafy stem. Uses floral bouquet, ornamental cut flowers. Peruvian lily, usually the flowers are beautiful, but they have an allergens, you see, which is called tuliposide A, the biosynthetic precursor of the allergenic lactone tulipalin A, has been detected in many species of Alstroemeria and Bomeria. Mirb is the scientist name. Alpha methylene gamma butrolactone is also present as an allergen in this flower. The allergy which caused is fingertip allergic contact dermatitis, is caused by contact with recent damaged plant and bulbs and gives rise to condition known as tulip fingers. Typically patients are florists who present with red, thickened and split tendered fingertips of both hands. Airborne contact dermatitis has been reported. This may reflect sesquiterpene lactone sensitivity. Astragalus is a very interesting plant which is having bulb looks like onion, garlic, they all comes in an underground bulb, onion you can say. Asparagus, common name asparagus, botanical name Asparagus officinalis, family, it belongs to the family Liliaceae. Even onion also, like tulips, onion, garlic, Liliaceae. Its stem, you can see the stem which has more proteinous, more vitamins, not protein, more vitamins are present. Perennial plant produces separate male and female plant for 20 years or more. The underground portion of the plant consists of stem, from which the edible spear grow upward. Each spring, the male plant yield the best spears. The allergen present in asparagus are: asparagus contain asparagine, coniferin, and glucoside and vanillin. And this, the bulb of asparagus, the allergen, however, appears to be 1,2,3 tritiane and 4 carboxylic acid, a sulfur containing growth inhibitor which appears to be present mainly in the early phase of growth season. And what is the allergy it causes? The allergy of the asparagus plant, this bulb plant, is well recognized in Germany, particularly among pickers who collectors young shoots, appears to be most allergenic. Usually it causes fingertip dermatitis. This includes contact, urticaria, swollen lips from eating asparagus, conjunctivitis, rhinitis, and asthma. Chamomile, a beautiful flower which is in our garden. Common name, sweet Roman chamomile, German chamomile, botanical name, Anthemis nobilis. Chamomillum nobile, Marticaria chamomilla, family belongs to Astraceae, or you can Compositae, family of this plant. It's an interesting plant having allergen. Description: 25 into 40 centimeter, ultra hardy, with fine cut pungent leaf, daisy like white flowers as shown in the picture. In the early to midsummer, the dry flowers are used to make chamomile tea. Oil from this flower is said to be help for stitching and the pains in the sides, among all other uses. Allergen present in chamomile. The allergens of chamomile include nobilis, sweet chamomile, it's also called as, and disacetylamine or disacetyl metaricarin. This is German chamomile will have this allergen. Both are sesquiterpenes. Now daffodil. This is also a beautiful funnel-like flower. Common daffodil, Narcissus, botanical name Narcissus, Pseudo-Narcissus, family they belong to larger family Amaryllidaceae. Description: strap-shaped leaves coming from the ground. You can see that it is coming from the ground like kana. Have inner trumpet, cup-shaped, cone surrounding petals surrounded by yellow, white, orange petals, usually with one flower per stem, very hardy. Uses ornamental flower, narcissus oil used in perfume manufacturing. It has allergy through beautiful daffodil have resulted in severe allergic problem including death. Symptoms have included allergenic rhino, conjunctivitis, asthma, allergic contact dermatitis, irritant contact dermatitis. Most patients affected work in flower industry, bulb, grows, stem, pickers, collectors, or florists who is working in their gardenist, but they are reported of reaction from contact with narcissus oil used in perfumes and fragrance. Narcissus contain a number of alkaloids including masonin and homolyscorin. This is the congestion and crystal of calcium oxalate seems to cause the characteristic source. Now fig, you know very but fig tree, fig fruit, common name fig in English, botanical name Ficus cariga, family Moraceaceae, mulberry family. Fig which is seen in our botanical garden of our college. The fig is picture delicious tree up to 15 meter tall, but more typically to the height 3 to 10 meter. They branch are muscular and twisted, spreading wider than they are tall. Edible fruit uses. The milk juice has been used as a destructive treatment for wart and to cure skin infections. The juice of fig leaves has long been used to treat vertigo, psoralen. Allergens present in fig: the fluorocumarin, psoralen, bergaptan, and the coumarin umbelliferan, 4,5-dihydropsoralen, and marmicin are present mostly in leaves and unripe fruits. The allergy caused due to presence of these allergens in fig: phototoxic. Ingestion of the fruit apparently does not cause photosensitization. However, there are reports of anaphylaxis after eating figs. Garlic, which we are commonly using garlic, we know this botanical name Allium sativum, family Liliaceae, uses for food. Garlic has a long history of being used in contemporary medicine to protect against cough and cold, and it is commonly used in poultice, which can cause very nasty burns. Allergens present is disulfide, allicin, and allergy caused probably the most common cause of allergy contact dermatitis on the fingertips. Most commonly it affects the thumbs, index and middle fingertips and non-dominant hands as a clove of garlic is held. Inhalation of garlic dust causes severe asthma. Garlic poultice can cause significant burns and probably irritant. Ginkgo biloba is one of the interesting leaf, and the plant is used for many purposes, and its resin is also. Botanical name Ginkgo biloba, family Ginkgogenaceae. The Ginkgo biloba is world oldest living tree, and allergen which is present is gingaolic acid. Allergy caused allergy contact to D tree. Food poisoning has occurred in Japan when too many nuts are eaten. It appears to be caused by MPN, that means 4-ortho methyl pyridoxine, which is present as an allergen and as an antivitamin, it is not a vitamin, antivitamin B6 activity. Kiwi fruit, which we have many dried fruit they used to supply in our, and fresh also available. By the common name kiwi fruit, zipri, Chinese gooseberry, kiwi berry, young tau, botanical name Actindia chinensis, family Actindiaceae. Uses used as in food, the vine and the great growing on Teliaceae long fence, covering walls or tall stumps. Allergens present, the main allergen present in the fruit is proteinase, actinidin. It is one of the group of the plant thiol, sulfur containing proteinase enzyme, to which papain, ficin and the stem bromelain also belong. Actinidin is composed of at least 2 proteolytic enzyme of increasing concern in the cross action between these proteolytic enzymes. And natural rubber latex. Individuals who have developed contact urticaria from exposure to the natural latex, usually from wearing rubber gloves, need to avoid eating and handling kiwi fruit. Lavender, you know that flavoring agent most of the time we are using. Common name lavender to wash, botanical name Lavender of Lavandula officinalis, family Lavandula, species Lamiaceae, uses ornamental plants, flower, narcissus oil used in perfume manufacture. Allergy, although beautiful daffodil, have resulting several allergenic problems including death. So symptoms have been included allergenic rhino, conjunctivitis, asthma, allergenic contact dermatitis, irritant contact dermatitis and contact with narcissus sap and urticaria. This narcissus oil is present even in daffodil. Lemon, which we are using commonly, common use lemon, botanical name citrus lemon, family Rutaceae, used as a food and garnishes. Lemon juice is added extensively in carbonated beverages, peas and tarts as a flavoring, and bakery, confectionery, preserves the pharmaceutical products. Lemon peel oil is used for furniture, polishes, detergents, soaps, shampoos. Lemon peel hydrated in the market of cattle field. But lemon juice has traditionally been used for diuretic, astringent and febrifuge, gingivitis and inflammation of the tongue. But the thing is, the lemon is also having allergen, psoralen, bergaptan, geraniol, citral, D-limonene. They also causes allergy. Citrus fruit, notably lemon and citrus bergamia, the source of bergamot oil, are important cause for phototoxic reaction. The peel of lemon is also irritant to cause contact urticaria. Allergic contact of dermatitis is unusual, although enhanced dermatitis has been reported in food handlers who had become sensitized by lemon. Citrus fruit peel is often waxed and dried. Sensitization may be due to carnauba wax or dyes such as carotene and citrus red 3 dye. Mint, which we know very commonly, peppermint mentha, mint, botanical name Mentha preparata, family Lamiaceae, uses ornamental plant, Narcissus oil used in perfume manufacturing, flavoring. Allergens menthol and L-carvone appears to be the main allergen. Ment also contain 1,8-cineol alpha penin. Allergy, although beautiful daffodil, have resulting of allergy problems including death. Symptoms include allergy, rhino, conjunctivitis, asthma, allergy, contact dermatitis, irritant contact dermatitis. These are some of the examples given. Students, just listen and write down. You will remember the examples for appearing the exam. Thank you. Thank you for listening.